Okay, so the final talk of the session will be by Damla Senol Cali. She's a staff software engineer at Bio-Nano Genomics, focusing on hardware acceleration. She received her PhD degree in computer engineering from the Safari Research Group at CMU in August 2021. She was advised by Professor Onur Mutlu and Professor Sigata. Her research interests are hardware software co-design for accelerating bioinformatic applications, genomic data analysis, and processing in memory. And today she'll be presenting her work on SeGraM, a universal genomics mapping accelerator, which is joint work with her collaborators at Carnegie Mellon, ETH Zurich, UIUC, Bill Gates University, and Intel Labs. Go ahead.
Can you hear me well? Yeah. Thank you so much for the introduction, Chris. Hi, everyone. Thank you so much for joining my talk. Today, I will be presenting our work, SeGraM. Genome sequencing, which enables us to determine the DNA sequence of an organism, plays a pivotal role in areas such as personalized medicine, outbreak tracing like COVID-19, and the understanding of evolution. So modern genome sequencing machines extract small randomized fragments of the original DNA sequence, which we call reads. And state-of-the-art sequencing machines either produce short reads or long reads. Read mapping, which is the process of mapping the reads to a reference genome, is a critical step in genome sequence analysis. And traditionally, we perform sequence-to-sequence mapping, where we map reads collected from an individual to a known linear reference genome. And this representation emphasizes genetic variations that are in the single reference genome and ignores all the other variations that could exist in the population, as you can see here. So this introduces reference bias, but as I said, this is a traditional way of performing read mapping, so it is a well-studied problem with many available tools and accelerators. So recent works replace the linear reference sequence with a graph-based representation of the reference genome, which we call as Genome Graphs. So with the existing of these Genome Graphs, we can map our reads to this graph representation of the reference genome. So this helps us to capture the genetic variations and diversity across many individuals in a population, so it results in notable quality improvements in genome sequence analysis. However, it's a more difficult computational problem with no prior hardware design. To this end, our goal is to come up with a specialized, high-performance, scalable, and also low-cost hardware algorithm co-design that alleviates bottlenecks in multiple steps of sequence-to-graph mapping. To this end, we propose SeGraM. SeGraM is the first universal algorithm hardware co-designed genomic mapping accelerator that can effectively and efficiently support both sequence-to-graph mapping and sequence-to-sequence mapping, which can be seen as a simpler variant of sequence-to-graph mapping for both short and long reads. We demonstrate that SeGraM provides significant improvements for multiple steps of the sequence-to-graph and sequence-to-sequence mapping pipelines. Let's look at genome graphs and sequence-to-graph mapping in detail. As we said, genome graphs are better suited for expressing the genomic regions that have variations than a linear reference sequence, since genome graphs combine the linear reference genome with the known genetic variations in the entire population as a graph-based data structure. Genome graphs enable us to remove the reference bias that using a single reference genome introduces, and therefore there's a growing trend towards using genome graphs to more accurately express the genetic diversity in a population. Let's look at an example. Let's say this is our starting linear sequence, so we can start with a single node, but let's assume in the population we have also this alternative sequence where on the fourth position we observe a G instead of a T, so there's a substitution. What we can do, we can divide this node into two nodes for representing the common subsequences, have one path to representing the first sequence and have another path for the second sequence. And let's say we have a third one. In this scenario, we have an insertion on the fifth position with a T. So we can have an additional node in our graph to represent this third sequence. And let's say we have a fourth one where that on the fourth position, the T doesn't exist at all, so there's a deletion. So we can have a direct path from the first node to the last node. So with increasing importance and usage of genome graphs, having accurate and efficient tools for mapping genomic sequences, the reads, to these genome graphs has become crucial. Which brings us to sequence to graph mapping pipeline. So this pipeline has 2 pre-processing and 3 main steps. And the first pre-processing step constructs a genome graph using a linear reference genome and the associated variations for that genome. And in the second pre-processing step, we index the nodes of the graph, and the resulting index is used in the first main step of the pipeline, which is seeding, which aims to find the seed matches between the query read and a region of the graph. After optionally filtering, clustering, or chaining these matches, alignment is performed between all of the non-filtered seed locations within the graph and the query read to find the optimal alignment. So alignment is the most expensive step of this pipeline, so let's look at it in detail. Traditional sequence-to-sequence alignment typically employs a dynamic programming-based algorithms with quadratic time and space complexity. A DP-based algorithm operates on a table, as you can see here, where each column of the table corresponds to a reference character, and each row of the table corresponds to a query read character. And in sequence-to-sequence alignment, a new cell, which you can see here, the dark blue one, the table is determined with simple rules from 3 of its neighbor cells. Whereas in contrast to sequence alignment, when we look at sequence to graph alignment, we have to incorporate non-neighboring characters, which are the hops actually, so which can be coming from non-neighboring cells. For example, here for the green shaded cell, we have to incorporate the 3 neighboring cells, but also 2 other non-neighboring cells as well. In order to further understand the performance bottlenecks of the state-of-the-art sequence to graph mapping tools, we rigorously analyze 2 such tools, Graph Aligner and VG, and we make 4 key observations. First, among the 3 online steps of the read mapping pipeline, alignment constitutes majority of the end-to-end execution, plus it is even more expensive than its counterpart in the sequence-to-sequence mapping pipeline. Second, this alignment step suffers from high cache misrates because we are generating and reusing a lot of internal data. And third, seeding step suffers from the DRAM latency bottleneck because we are doing a lot of irregular memory accesses to the main memory. And fourth, both of the tools scale sublinearly. So because these problems exist, we also look at hardware accelerators, but as I said, there's no hardware accelerators specialized for sequence to graph mapping. So we looked at existing sequence to sequence mapping accelerators, but they are unsuitable for the sequence to graph mapping problem because they cannot handle the graph-based inputs, the reference genome. So we looked at existing graph accelerators because we have many of them, but they are promising for solving the seeding problem when querying the main memory, but they are unable to handle the actual bottleneck problem, which is alignment, because alignment is not a graph traversal workload. So we need to have a specialized, balanced, and scalable design for compute units, on-chip memory, and also main memory accesses for both the seeding and alignment steps of sequence to graph mapping. To this end, we propose SeGraM. SeGraM is the first universal genomic mapping accelerator that can support both sequence to graph mapping and sequence to sequence mapping for both short and long reads. And to our knowledge, SeGraM is the first algorithm hardware co-design for accelerating sequence to graph mapping. And we base SeGraM upon a minimizer-based seeding algorithm, and we propose a novel bit vector-based sequence to graph alignment algorithm. So these are our algorithmic contributions. We co-designed both algorithms with high performance, scalable, and efficient hardware accelerators, and this covers the hardware level contribution of SeGraM. So when we look at the SeGraM hardware design, it consists of 2 main components. First, MinSeed, which is responsible for the seeding step, and it is the first hardware accelerator for minimizer-based seeding. And second component, BitAlign, is responsible for the alignment step, and it is the first hardware accelerator for sequence to graph alignment. Let's look at the high-level overview of SeGraM. So before SeGraM execution starts, as pre-processing steps, we generate each chromosome's graph structure, index each graph's nodes, and then preload both the resulting graph and the hash table-based index into the main memory. So both of these data structures are static, so we can generate them only once and reuse for multiple mapping executions. So let's look at the overflow. So SeGraM execution starts when the query read is streamed from the host and MinSeed writes it to its scratchpad. And using all of the k-length subsequences, which are called k-mers of the query read, MinSeed finds the minimum representative set of these k-mers, which are called minimizers, according to a scoring mechanism, and writes them to the minimizer scratchpad. And for each minimizer, MinSeed fetches its occurrence frequency from the hash table sitting in the main memory and filters out each minimizer whose frequency is above a user-defined threshold. And here we aim to select the least frequent minimizer so that we can decrease the burden on the alignment step. Next, MinSeed fetches the seed locations of the remaining minimizers from the main memory and writes them to the seed scratchpad. And finally, MinSeed calculates the candidate reference region, which is the subgraph surrounding the seed. For each seed, fetches the graph nodes from the main memory and writes the nodes to the input scratchpad of BitAlign. And then BitAlign starts by reading the subgraph and the query read from the input scratchpad and generates the bit vectors required for the alignment operation. So while generating these bit vectors, BitAlign writes them to the hop queues in order to handle the hops required for graph-based alignment, and also to the bit vector scratchpad to be later used as part of the traceback operation. And once BitAlign finishes generating and writing all these bit vectors, it starts reading them back from the bit vector scratchpad, performs a traceback operation, finds the optimal alignment between the subgraph and the query read, and streams the optimal alignment information back to the host. When we look at the hardware design of MinSeed, it consists of 3 computation modules, 3 scratchpads, and the memory interface. So the computation modules are implemented with simple logic since we require only basic operations like comparisons, simple arithmetic operations. And for all the 3 scratchpads, we employ a double buffering technique to hide the latency of the MinSeed accelerator. And we also couple MinSeed with high bandwidth memory to enable low latency and highly parallel memory access. When we look at the hardware design of BitAlign, we implement it as a linear cyclic systolic array-based accelerator. So while this design is based on the GNASM hardware, where bit vectors are written to bit vector scratchpads during bit vector generation step for later use in traceback, our new design incorporates hop queue registers in order to feed the bit vectors of non-neighboring characters or nodes, so the hops. And when we look at the overall system design of SeGraM, it is connected to a host system that's responsible for the pre-processing steps and for transferring the query read to the accelerator. And next to each HBM channel, we place one SeGraM accelerator such that it will have exclusive access without any interference from other SeGraM accelerators. And each SeGraM accelerator is a combination of one MinSeed accelerator and one BitAlign accelerator. So each HBM stack has 8 memory channels, so connected to 8 SeGraM accelerators, which define one SeGraM module. And our hardware platform includes 4 off-chip HBM stacks. So we have 4 SeGraM modules in total, so 32 SeGraM accelerators in total. As a result of the flexibility and modularity of the SeGraM framework, we can run each accelerator together or separately. So we describe 4 use cases of SeGraM, and the first one is end-to-end mapping with SeGraM, where we need to run MinSeed and BitAlign together. Second, we can have sequence to graph alignment as our use case, where BitAlign will be standalone, but it can be coupled with any seeding alignment, seeding tool or accelerator. Similarly, we can use it for sequence to sequence alignment. And our 4 cases, use cases seeding for both linear or graph-based mapping, where we will be running standalone MinSeed, but it can be coupled with any alignment tool or accelerator. Let's look at our evolution of the SeGraM design. So I won't be able to go into details of our methodology, but you can refer to our paper or we can talk offline. But based on our synthesis of the MinSeed and BitAlign Accelerator datapads, total area overhead of SeGraM attached to all 32 channels is around 28 millimeters square, and the total power consumption is around 28 watts, including the HBM power. And we find that main contributors for the area overhead and power consumption are HOP-Q registers, since they constitute more than 60% of the area and power of BitAlign logic, and also the bit vector scratch pads. Let's look at our long-read analysis for sequence to graph mapping with SeGraM. Here we only show our throughput numbers in the plot, not the power numbers. So we compare SeGraM with two state-of-the-art sequence to graph mapping software tools, Graph Aligner and VG. And we find that SeGraM provides around 6 times and 4 times throughput improvement over these baselines, while reducing the power consumption by more than 4 times for both. And with the exact same baselines, but this time with short reads, we find that SeGraM provides even higher amount of throughput improvement over both of these baselines, while reducing the power consumption by, again, more than 3 times for both. And let's look at our key results for the sequence to graph alignment use case, where we are running BitAlign as a standalone, and we compare it with software-based tool Pascal. And we find that depending on various data sets, as you can see here, BitAlign provides between 4 times to more than 500 times speed up over Pascal. And as we said, BitAlign can also be used for sequence-to-sequence alignment, but the cost of more functionality is extra HOP-Q registers in order to support sequence-to-graph alignment as well, but we don't sacrifice any performance for either use cases. So compared to state-of-the-art hardware accelerators, but this time specifically designed for sequence-to-sequence alignment, we find that BitAlign increases throughput for over all 3 baselines, but with the extra cost of power consumption and area overhead. So before I conclude, here's a list of additional details that you can find in our paper. For example, sources of improvements in SeGraM. So to conclude, in this work, we propose SeGraM, the first universal algorithm hardware co-designed genomic mapping accelerator that supports both sequence-to-graph mapping and sequence-to-sequence mapping for both short and long reads. SeGraM consists of 2 components, MinSeed and BitAlign, and it supports multiple steps of sequence-to-graph and sequence-to-sequence mapping pipelines. And based on our extensive analysis, we show that SeGraM outperforms state-of-the-art software and hardware solutions. So our paper is available online, and we will be releasing our software implementations and the data sets we use in our GitHub page soon. Thank you so much.
We got time for a question or two. Maybe one question about the HOP-Q. So it seems like one of the key differences between sequence to sequence and sequence to graph is these additional edges. And I think those are tracked by the HOP-Qs. It seems like the HOP-Qs would have to be quite large to capture very long hops. So how do you navigate that trade-off?
That's a really good question. So there is a trade-off there. We cannot, of course, the graph can be huge, even though we are generating them per chromosome. But in SeGraM design, we limit our HOP-Q register size as 12 based on our empirical analysis, because when we do that, we found that we cover more than 99% of all the hops exist in the generated graphs. So we lose some hops with this design choice, but we save significant amount of area and also power as well.
Okay, go ahead. Yep.
Yeah, thank you for this talk. Just a quick question.
Don't forget to introduce yourself.
So is there a specific reason why you choose to use HBM? And the second part is, if we want to use this approach, which let's say standard DRAM, how will you do?
So could you please repeat?
He's asking about HBM?
Yeah.
Specific
reason to choose to use HBM, and then the next part of the question is, if we want to use standard DRAM, how?
Right. That's a really good question too. So the reason why we pick HBM is just to have exclusive access for a portion of the memory. So they won't be interfered, the multiple accelerators won't be interference with each other, but the high band, like memory bandwidth requirement is not that high, so we are not exploiting that. So which kind of answers your second question. We can definitely attach a typical DDR kind of memory to SeGraM. The only thing we have to add on top of is the memory access management. That's it. Yeah, thank you.
We get time for one more question.
Hi, great paper. Just wondering, what were the read sizes like for long reads that you were looking at, like N50 or something?
Yeah, 10,000 we picked. They are simulated reads.
Okay. How much would you how would you think the performance is going to change? Like, is there like some sort of a read size limit that would affect the performance?
Yeah, so that's a good one too. So we are following a divide and conquer approach where we are dividing our inputs into chunks so that they won't be dependent on our mechanism won't be dependent on the inputs. So it will scale linearly because just the number of windows will increase in that scenario.
Right. And for the read errors, what's the percentage at which you feel like that the performance is like not good?
So we picked 5% because the technologies are around that bar these days. So when we look at different error profiles, again, it is just a constant with our this approach in our underlying algorithm and also the mapped accelerator as well. So
thank you.
Thank you.
All right, great. So let's go ahead and thank Damla. And this does conclude this session. So I think now we have a break.